Hello everyone, just look at this image. At first glance, it looks complicated, messy, and impossible to remember. But this is the most beautifully organized system in the human body. If you try to memorize each pathway separately, you will not understand it. But if you understand how they connect with each other, everything becomes very simple. First, always look for the center of metabolism. As you can see here, this is the center. The center of metabolism is the Krebs cycle. It is the metabolic hub where everything meets. So let us simplify this image into this image. So this is the interconnected metabolism pathways. So all the pathways, whatever we eat, either it can be carbohydrates, fats, proteins, everything will meet at the Krebs cycle. So for instance, glucose will be converted into pyruvate by glycolysis. This pyruvate will be converted to acetyl-CoA, which will be entering the Krebs cycle. This pyruvate to acetyl-CoA conversion is pyruvate dehydrogenase enzyme, which acts and converts the pyruvate into acetyl-CoA. So coming to the fats undergo beta oxidation and converts to acetyl-CoA, which enters the Krebs cycle. The proteins are way more simpler. They can enter either in the form of pyruvate, acetyl-CoA, or Krebs cycle intermediates and enters the Krebs cycle and produce the carriers, which are NADH and FADH2. So from here, these will enter the electron transport chain and will produce the ATP, which is the energy. Ultimately, whatever we eat is to produce the energy and do the work. So that's what's happening. So not only the things. In protein metabolism, there is a production of ammonia, which is toxic to the body. We have to remove that also, right? So that's why urea cycle takes place. The ammonia will be converted to urea. In between these, there are substrates like aspartate and fumarate which are formed. These will re-enter the Krebs cycle and produce the energy. So even the waste management and also the energy management is also interconnected. So the energy and the waste pathways are also interconnected. So let us see in this image, we can see the glycolysis, the protein metabolism pathway, and even the all these things will enter the Krebs cycle. The proteins will enter the glycolysis, the glucose, carbohydrates will enter. For the fat metabolism, the beta oxidation, the fats will enter. Finally, they convert to acetyl-CoA and they enter the electron transport chain. From there, the NADH will be converted to ATP and ADP, and we use that energy. So, what happens? Let's see the clinical correlation. What happens if this not occurs? In thiamine deficiency, thiamine is an important thing for the pyruvate dehydrogenase, which converts the pyruvate into acetyl-CoA. If there is deficiency in that, no acetyl-CoA production, there is pyruvate will be converted to lactate instead of acetyl-CoA. More lactate will cause lactic acidosis and neurological problems. In diabetes, there is excess acetyl-CoA. Excess acetyl-CoA will be converted to ketones instead. So this causes ketoacidosis. So in cyanide poisoning, what happens is the electron transport chain, up till here it's normal. The ETC will be blocked in cyanide poisoning. The NADH will accumulate. As the NADH is accumulating, feedback, it will inhibit the Krebs cycle. So there is no ATP production. So, and it is toxic to cells. That's why. In MCAD deficiency, what happens? It cannot form the acetyl-CoA. There is no energy during the fasting. That's why the fats cannot be converted to acetyl-CoA. So that's why there is hypoglycemia without ketosis. What happens in hyperammonemia? The urea cycle will fail. The ammonia will accumulate. That is toxic to brain and reduces the Krebs cycle activity. So in one block in metabolism affects multiple pathways at once. That's why biochemistry is not about memorizing the pathways. We should understand the connections, the interlinking between each other. So the glycolysis, any pathway which enters the Krebs cycle, it will enter the ETC, electron transport chain, and produce the ATP. In thiamine deficiency, there is no acetyl-CoA, lactates increase. diabetes mellitus, there is in excess of acetyl-CoA which converts to ketone and ketoacidosis. In cyanide toxicity, there is ETC which is inhibited and no energy production, toxic to cells. And if there is hyperammonemia, this will be inhibited because the urea cycle fails, the ammonia accumulates. So that's why it reduces the Krebs cyclic activity. So all the pathways are interconnected. So it's all about understanding the connections between the pathways.